Good afternoon. I'm going to speak on the potential of soil cyanobacteria to restore soil in dry lands. This is a project we are developing at the University of Almería in Spain in collaboration with other universities in Spain, Italy and Australia. We already know that the difficulty to restore soils in dry lands because of the harsh environmental conditions under which traditional approaches based on the establishment of a plant cover have poor results. On the contrary, biocrust or biological soil crust, which are complex communities of microalgae, fungi, cyanobacteria, lichens and mosses, can adapt themselves to these extreme environmental conditions. And furthermore, they play very important functional roles, especially on soil-water balance and on carbon and nitrogen cycles, and also reducing erosion. So, biocrust restoration seems to be a promising strategy in assisting landscape restoration in dry lands. Thus, the cultivation of biocrust components in the lab is starting to be used to reestablish them in degraded areas. Among the different biocrust components, cyanobacteria are very interesting because they are the first colonizers after a disturbance. And they also tolerate very extreme environmental conditions. They are able to fix carbon and to secrete exopolysaccharides which favor the availability of water in soil surface. And also they act aggregating soil particles, reducing soil erosion. Some species are also able to fix nitrogen, and with this they increase soil fertility. Moreover, cyanobacteria facilitate the succession of lichens and mosses and later plants. And strains of interest can be selected, isolated, and cultivated in situ to produce the needed biomass of cyanobacteria at low cost and on short period of time to face large-scale restoration projects. In light of that, the aim of our project is to demonstrate that cyanobacterial inoculation or cyanobacterisation is an effective strategy to reinstate soil function in large-scale rehabilitation. To achieve it, we have been working in three specific objectives. The first step was to select appropriate native cyanobacteria strains and to analyze the effects of cyanobacterial inoculation on different types of soil under laboratory conditions. The second step was, or our second objective was to optimize the production of these strains to face large-scale restoration, and we are currently working on applying different strategies to enhance cyanobacterial colonization under field conditions and evaluating their effects on soil properties. We are working in three representative ecosystems in the southeast of Spain, in the province of Almería. All of them have a thermo-Mediterranean semi-arid climate and are subjected to different degradation processes and have different soil textures. Specifically, we are working in a limestone quarry, in the woodland systems, and the lowest degraded, which is a steep ecosystem under grazing. We selected three nitrogen-fixing strains, and they were chosen because of their functional capabilities, because they are very characteristics of these sites, and also because they were easy to isolate and grow. We did the second part of our first objective to analyze the effects of cyanobacterial inoculation on different types of soils under laboratory conditions. We grow the strains in the laboratory to obtain the needed biomass to do our experiments, and soil samples of the 3 sites were inoculated with each strain individually and also with a mix of the 3 strains. And we monitor the colonization of the cyanobacteria. For that, we measure the cyanobacteria coverage. Also, we measure the spectral response of soil surface, and at the end of the experiment, we took samples and we determined chlorophyll content and also soil organic carbon and total nitrogen content. We found that all the strains were able to colonize the different soils and improve significantly soil properties respect non-inoculated soils. Nostoc commune and the mix of the 3 strains promoted the highest improvements in soil properties. Looking at the cyanobacteria cover, we found that at the end of the experiment, I didn't say that the end of the experiment was only 3 months. At the end of the experiment, we showed the 3 soils showed higher cover of cyanobacteria, respect the non-inoculated soils, obviously. And in all cases, we can see that Nostoc and the mix of the 3 strains reach the higher values of cover. The colonization reached values of 50% in the less degraded area, las amoladeras. Sorry. This is the less degraded area. So here we can see, in fact, in all cases, both options, the Nostoc and the mixture, were the best options. Regarding the effects on soil properties, we found that all inoculated soils at the end of the experiment showed soil organic carbon gains. In contrast to the controls, the non-inoculated controls. And the highest organic carbon gains occurred in the most degraded area. Nitrogen gains, where the pattern of nitrogen gains was very similar to this of soil organic carbon. Then our second objective was to optimize the production of these strains to face large-scale restoration projects. For that, apart from making use of sunlight to produce the biomass, we have been working on reducing the cost of media to culture the cyanobacteria. So we have been using low-cost agricultural fertilizers to buy our culture media. And we have analyzed the growth of our 3 strains in reactors with culture media made by low-cost fertilizers, and we compared with media made by chemical compounds. Also, to evaluate the quality of these cultures, of this inoculum, we inoculated them on the soil of the quarry. Previously, we sterilized the soil. And we followed the colonization of cyanobacteria and the effects on soil properties. In the figure, we can see the growth evolution of Nostoc in the standard medium VG level with nitrogens, with chemical compounds. And in dash, the growth evolution of the same species, of the same strain, in a medium with the same composition of VG11, but made by fertilizers. We can see that they are quite similar, the models are quite similar, and also for the other strain, Tolipotrix, and even for Citonema, our third strain, it was even better. So the culture with fertilizers allowed an evolution, growth rates, and total biomass production quite similar to the media made by chemicals. To see the quality of our inoculum, we inoculated the soil, and we found that the culture made with fertilizers reach similar colonization rates than the cultures made with chemical compounds. In fact, they promoted higher soil organic carbon content and EPS production. This is for nostoc. The behavior was very similar for the 3 strain. This is the best, obviously. So we are able to produce a low-cost inoculum in a short period of time, and we are able to face large-scale restoration in the field. The next question is, does it work under field conditions? Okay, our first inoculation was carried out in winter, looking for rainfalls, because we have 200 millimeters of annual rainfall in these sites. We inoculated in the 3 sites the mix of the 3 strains. A week after the inoculation, a very extreme event occurred in our 3 sites, which is typical, is a characteristic of our weather there. And all the event detects and removes all the inoculated biomass. So we repeated the inoculation at the end of the spring, avoiding the rainfalls. But at the end of summer, after a very hot and windy summer, the colonization rates were really similar to the natural colonization rates. We are actually working on applying different strategies to reduce the environmental stress in order to facilitate the colonization of the cyanobacteria. Our preliminary results, but we are testing, for example, different type of mesh, covering the inoculated plot with different types of mesh. Our preliminary results show that after 6 months of inoculation, chlorophyll content, which is an indicator of the colonization of cyanobacteria, is much higher in those plots inoculated and covered with the vegetal fiber mesh. So it seems to be a promise, I don't know. We will see. We can conclude that soil inoculation with cyanobacteria can be a cheap and efficient tool for restoring soil function in degraded drylands if strategies to reduce environmental stress are applied to enhance the inoculum survival under field conditions. Thank you very much for your attention.
Stabilization, do you have a way of, I guess, field-proofing your field site to try and, because you've removed the vegetation and layers or just spray down the cyanobacteria, that you've created an erosion zone. Is there a way to sort of barricade to try and stop that erosion?
Yes, we can apply the traditional strategies to stabilize the soil, but we were looking for a direct, cheap, interesting strategy, because they really in the field they appear just after an event, and they are able to survive in very extreme conditions. So we thought that after inoculation it will work, but it doesn't work. Not only for us, for the rest of the scientific community, we are collaborating with different groups. They have the same problems, but it seems that these strategies to stabilize the soil. Especially from wind. We think that wind is the most dangerous. Maybe will work. Thank you. Thank you.
So next we have Professor David Eldridge, who will be talking about harnessing the activities of native animals to restore degraded environments.
Beautiful. Hi everybody, thanks for coming along. This is probably a little bit kind of off cue for most restoration meetings, but I've been pretty excited about what animals do to soils and how important they are for potentially restoring degraded systems. Now I work in drylands, the sorts of environments that Yolanda showed us. Very harsh, high evapotranspiration, low rainfall, seasonally variable. And these are the sorts of restoration projects that you see. Very broad scale, very expensive, high cost, high energy. This is on the Riverine Plain in eastern Australia where they've planted saltbush. Even more high energy engineering approaches, these ponding banks west of Nyngan in central New South Wales. Very, very effective. They control water movement. Water goes through the profile, leaches salt, germinates seeds, and you get this amazing germination. Very, very expensive. Something that the Western Australians, because a lot of Western Australians here at this workshop, at this conference, have pioneered is a process called tine pitting, where a machine goes through the soil, it's 3 tines, they're on a cam, they move up and down, and they create these series of pits. And when I looked at these pits, I thought, these pits are really similar to the sorts of pits that echidnas and bilbies and betons, the stick-nest rats, and rabbits, and a lot of these soil-disturbing animals create. I wonder how these animals disturbing soils might produce a similar result as we get from a very expensive technique such as tine pitting. Why echidnas? Well, echidnas are pretty much everywhere. I'm sorry I don't have a map of Australia, but you can see this is a study of one of my colleagues, Dan Lundy, did, a postal survey, and basically he showed that echidnas were all over the state. Now they're a bit sparse out here because these represent individual landholders on large properties, so echidnas are pretty much everywhere. But they don't forage randomly. They dig a hole when they're foraging for termites or ants, and their foraging is non-random, so they'll dig a hole under trees, under logs, under shrubs, under grasses. So this patch in here represents a patch of trees with shrubs. That line is 50 metres by 4 metres. So this is where we've been monitoring the effects of different animals on soils for a number of years. So we thought we'd set up an echidna experiment. Now, we didn't want to actually go to pits constructed by echidnas because, of course, they're non-random. We wanted to have a randomised block design where we could select pits where we wanted them to. So we created our own pits. And when you've counted as many echidna pits as I have, probably tens of thousands over the last few years, you'll know what they look like. So we constructed these pits. We had a transect of about 1 kilometre long. Along that 1 kilometre long transect, we had 20 stations or blocks. Each station had 2 pits and 2 controls or non-pits. And here's an example of a pit here. We did a whole bunch of measurements over 18 months. We measured things such as how long it takes for the pit to fill in, how they capture seed and litter, decomposition and labile carbon. So what did we find? Well, we found that pits of echidnas trapped about 5 times as much litter as the surface. And when you're in these environments, and I know Yolanda would sort of would recognise an environment like this, the interspaces are almost bare. There's pretty much nothing there unless you've got an obstruction. And that's what the pits are providing. They're providing an obstruction where the wind changes and material drops into the pit. Now, because material is dropping into the pits, they lose their structure very rapidly. And over 18 months, they almost become, well, not 18 months, maybe 24 months, they become almost indistinguishable from the surface soil. And this is pretty much consistent with what we know about animals digging a range of structures, that the longevity of the structure is proportional to the size of the structure that they're producing. So they fill in relatively quickly. Now, with our 2 pits and 2 controls, we did a litter decomposition study. So we put litter of Eucalyptus intertexta into a litter bag. We put them both in the pits. We covered one with soil. We left one uncovered, and we looked at decomposition. Well, it didn't really matter whether you covered it or not, because within about a month, everything was covered. So what we found at the end of the experiment was that there was about 15% greater decomposition in the pits, irrespective of whether we put soil in them or not, compared to the control. So litter is decomposing much more quickly in the pits, and we've done a lot more work on different animal pits looking at things like decomposition. So they're catching more soil, they're catching more litter, they're actually catching more seed. Now, I don't have data here on the seeds. It was an interesting study because we would go out every time, we'd take all the litter out of the pits, we'd go back, we'd sort through all the litter, take out all the seeds, put the seeds back in, take it all back, put it back in the pit. Did that for 18 months. What we found was that we had about 4 times as many plants coming up in the pit and about twice the richness of plants. Interestingly enough, the plants growing in the pits do not represent a unique assemblage of plants. They're pretty much just a subsample of what's in the environment. It'd be really interesting to know whether, with the loss of many of these soil-disturbing animals, whether we've lost a subsample of plants that would naturally occur there. And finally, thinking about function, our measure of function was labile carbon, which is that really active form of carbon that's made up of microbes, made up of soluble polysaccharides and things like that. Labile carbon is highly correlated with function. We could see that the red line is the control, so it didn't change much over time. A huge drop in labile carbon when you dig the pit, and that's because you remove Yolanda's cyanobacteria, you remove a whole bunch of the surface. Remember, all the action is in the first couple of millimetres in these arid soils, but eventually after about 18 months, labile carbon increases. And in fact, if we follow this, and I haven't plotted that there, the labile carbon is substantially higher in the pits, even though they're indistinguishable from the surface soil. So what these animals are doing is they're building up fertile patches in essentially a fairly infertile environment. So we can kind of put it all together, and this is what's called a structural equation model, but we don't really need to know that. All we need to know is that a red line is a negative relationship, and a black line is a positive relationship, and a thick line is a stronger relationship. So we can sort of have a look at some of the mechanisms. So if we're thinking about germinants, thinking about getting plants to grow, we know that pits catch more seed, and that creates more germinants. But we also have this negative effect here, which we have to balance out with a positive effect, in that more litter from the pits results in fewer germinants. And I'm not exactly sure what that relationship is. Maybe it's a smothering effect. I'm not exactly sure. And down the bottom here we have things like pits have more litter, which suppresses labile carbon, and then some direct effects. So we can start to tease apart some of the mechanisms why these pits might be important. What about management? I have 2 hats. I work for the Office of Environment and Heritage. And they're always asking me, oh, well, this sort of work, this bullshit work is not very applied. It's pretty esoteric. You work at a university. I say, well, no, not really. Let's have a look at where we find echidnas. And this is a study we did in Western New South Wales in a functional woodland with lots of shrubs, lots of understory, really good grasses, biologically very rich, and an adjacent site which it still had the upper story, but it lost all its understory. Almost 14,000 foraging pits per hectare, compared with less than 300 foraging pits per hectare over here. So there's very many orders of magnitude more pits, and as a result, echidnas are digging something like 18 tonnes of soil here and 1 tonne of soil over here. So they're digging much more. The thing that we found really interesting is that when systems become degraded, the pits become much larger, which is really interesting because they're forced to dig deeper to change their change from ants to termites. So when systems become degraded, they're producing pits which are much greater. Maybe that's a positive feedback mechanism for catching more litter to restore the system. You can only sort of think about that. So how do we get the message out? Well, hopefully I'm getting the message out to you that not only are these animals important from their intrinsic biodiversity value, but they're critical for their engineering role. To increase echidnas, we need to restore habitat, and to do that, we need to enhance habitat connectivity. And finally, I know they're pretty prickly, but they are susceptible to foxes and things like that, so we need to control feral animals. A lot of this work comes from fantastic students that I've had over the last 10 years, which I still have now, and I think I'll leave it at that. Thank you. I understand where you're going. Yes. They cannot create litter, but the interesting thing is that litter on the surface is susceptible to breakdown by UV light, which means that the carbon doesn't get into the soil. These guys are bringing the litter into a patch, soil gets stuffed on top, and that's where all the action is. So I think that's the mechanism. You know, they're not creating more litter. They're just helping to harness it and to keep it underground where microbes and things get into it.
Did you consider the positive impact that fishing would have on soil infiltration and available water? Did you measure doing measurements?
We've done a lot of work on impacts of pit soil infiltration. You know, it's like when they dig a pit, they remove the biological crust, which, as we've seen from Yolanda's work, is really important. Crusts generally shed water, but the water's not lost. It's moving into another part of the landscape. So by digging pits, you'll get more water. I mean, the work from Western Australia, what's his name, Geochemical showed that you get greater soil moisture. So they're changing the distribution of water, and we can measure that, but I don't know what it means. You know, it's kind of at the landscape scale. Who knows? It's a big question.
I found in WA that it helps reduce water repellency.
That's right. Yeah.
So soil layer is actually stopping infiltration of water. And once they do that soil turnover and digging, any seedlings or seeds that are in that space actually have a better access to water and more ability to germinate. So we're doing some studies where they're doing diggings to replicate Wendy or Bounty Creek that we have in Southwest.
Yeah.
Is there another link in this in that you said they don't dig the holes around them, so that they make our carbon and everything else different in the places before they dig the pits?
Well, I guess what they're doing is reinforcing that frozen-off patchiness, that sort of spatial patchiness that exists in parasites. They do dig in the open, but they tend to concentrate it in the patches. So you're probably right. I mean, there are other things we haven't looked at for sure. Thanks.
So next we have Dr. Songlin Wu, who's from the University of Queensland. He'll be talking about geochemical and mineralogical constraints in the iron ore tailings and limit of soil formation for direct phytostabilisation. Thank you.
Thank you very much for the opportunity for making this presentation. Today I will give a talk on the constraints of the iron ore mine tailings. So from the perspectives of geochemical and mineralogical. So my talk mainly on these several aspects. First there is the background. My activity is one of the most important activities for our human beings. Because when you take a flight, you find there's vast areas of the land that was destroyed by the mine activities, and especially there's huge amounts of the tailings was produced. So this tailings has extremely chemical, geological, some traits that can not support the survival for microbes in the plants. So the Australia is a very bigger country that is nearly more than 28% of the world's iron ore reservation. And each year, millions of tons of tailings have been produced. So it's a big issue that's faced by the human activities by the government. And so traditional usually we use some soil, artificial soil to cover these tailings and to support the plant rehabilitation. However, these activities can cost a lot because when you dig a hole and get soil resources from other places, so this can cause some affecting the environmental impacts. So what we're going to do is we have a new method is to eco-engineer these tailings into some techniques. So our goal is to first to transform this to some soil-like materials that can support some initial peony plants to survive. And then second, we use these peony plants to accelerate the mineralizing and so improve some physicochemical trees, and then for some soil structure formations, especially the aggregation particles. This is very important for the soil structures. However, before we do anything, so we need to have a look at what's the constraints of these of the tailings. So then we can, based on what we know, we can get some technologies, develop some technologies to do the eco-engineering. So our question is, so first, what's the physical chemical characteristics of the iron tailings? The second is because it's iron tailings, there's the majority is iron being minerals. So what's the forms of the iron being minerals? And third is there some peony plants that which can survive on one side, which can make some difference on the tailing physical chemical and mineralogies. So our game is to investigate the physical chemical iron mineral phases speciation as well as enriched mortgage to know details on the constraints of the tailings. So based on these questions, we have sampled the fresh tailings, and we sampled on the one side is for 4 years without any eco-engineering course. And also we sampled the topsoil covering the tailings for 4 years, and there's also some tailings plants on the side. And also we collect one native soils, and for this we can compare the different traits of the tailings and the soils. So from the colors you can find the iron tailings is dark, and the Asian tailings there's some black, and between the black and the white, the topsoil is white. So let's look at the chemical trace pH. So you find the first tailings has a very high pH, and the soils is with a little acid. And also you can find the tailings as with a little high salinity. And we found that the soils has more organomatters. So the tailings, there's a leak of organomatter for tailings. So we have details on the organic forms. We use FTR. We further found that in the soils there's some carboxylic and some aromatic carb forms in the soils. There's the leak of initial abundant organic carbon in the tailings. And further, we have looked at the physical structures of the tailings and the soils. Especially this black is the macroaggregates percentage. you can find the soils has a higher percentage of the microaggregates, where the tailings leaks some aggregation structures. And especially we found in the minor surface areas found in the soils, there are higher PET surface areas, sometimes about nearly 10 times more than the fresh tailings. So we further detect the iron speciation change in the tailings and in the soils, especially this is chemical extraction methods. The AO iron is represented to some most amorphous iron and also some iron oxides like magnetite. And CVD represents most crystalline iron minerals. So you can find in the aged tailings, there's more AO and CVD extracted iron, and which is higher than the fresh tailings and native soils. And so this indicates that in the nature sites, the aging process can increase the iron oxide hydroxides produced. And at the same time, we found that in the soils, there's more aluminum, extracted aluminum. So there's a leak of aluminum in the fresh tailings and in the aged tailings. And here we use XRD, we further do the mineralogy of the tailings soils. And it's similar with the chemical extraction results. We found, in fact, the iron oxide hydroxide will increase, especially the hematite will increase during the aging processes. And also we found new is that in the soils there's a phytosilicates, but in the tailings there's no such phytosilicates. Usually the phytosilicates is very important for soil structure formations. And then we further detect the iron speciation between the tailings and the soils. Generally, from most of the iron in the tailings and the soils were in the iron 3 species, while in the fresh tailings there's a little in the straight shoulders there's a little iron 2. And especially from in the fresh tailings, they take a little similar like the biota. However, in the aged tailings and the soils, there's no typical structures of the biota yet, but they all similar with some fixed features of the oxide hydroxides. And further, the faking work found that, especially in the fresh tailings, there's more than 50% of the iron was reduced to biota-like minerals. While in the aged tailings, this biota was transformed to more oxide hydroxides, mostly is the hematite. In the soils, there again has some more geochemical minerals. So geosite is with high surface areas and the secondary iron minerals, which has more capability to interact with organic matters, and thus facilitates some more microaggregation formations. And further, previously we found the iron oxide hydroxides in the tailings. It's very important in the soil formation. However, these iron minerals usually should be interacted with some other minerals, such as some aluminum and silicate-rich minerals. So this form can take some part for association with organic matters, and this is the nature part. However, when we look at the tailings, we found, in fact, these iron-rich minerals with the small areas, and they were separated from some other minerals. Although in the aged tailings, we found there's some signature of drylands, and you can see this drylands only exists on some rather spills of the native plants. However, they were still distracted with other minerals and with sharp edges. And you can see here we collect some, this is very special native peony plants that can survive on the aged tailings. It's Mariana brachiaria. So So we did some tailings on the surface roots. We found, in fact, there's signal of minor one. So this indicates that initially these plants have great potential to enhance our mineral resin. So this can be useful for our further eco engineering course. Okay, let's give a summary on the key findings of our research. The first is our tailings are outlined with low organic matters and poor physical structures, especially poor aggregation, water-stable aggregation. And second, nature aging can alter physicochemical traces, but the pH is still is a little high. But they can initially biota the resin into some facilitate some formation of R-oxyhydroxides. And third, although the native aging can increase the R-oxyhydroxide production, but these R minerals are still in sharp edges and they still need of forward interaction with other minerals for aggregation. And lastly, there's some signature that some peony plants maybe can be advocated to use in the future eco-engineering course to accelerate some mineral resin or mineral association, so for structure formation. And generally, here is the summary of the whole findings. So the tailings is alkaline new organizers, low stable aggregation, and rich in primary minerals. And in the native aging result eco-engineering course, there are still alkaline and new organizers and low stable aggregation. Although there's some signal of mineral resin, but still rich in sharp aging minerals and nickel contaction between the R minerals with other acidic and aluminum minerals. And overall, we propose that the eco-engineering course is urgently needed to accelerate the mineral resin and to increase the organic matter accumulation and to facilitate the neutralization and increase the aggregation processes and thus facilitate the soil structure development for plant and for ecology restoration of the mines activities. Okay, report covers thanks for Professor Mihong for the help on the field work and thanks for the ARC and thanks for some Taiwan Central and also thanks for the CMM here for the SEM and stream analysis. Thank you very much.
So next we have PhD candidate Vicky McGimpsey, who's from the University of Melbourne. Thank you.
Hi, I'm Vicky McGimpsey. Today I'm going to be presenting A Question of Time, Microbial Community Change Along a Restoration Trajectory. I had to change the title a little bit because my mycorrhizal data only just came back, and there's sequences in the 10,000 order of magnitude, and I've got 5 different regions, so the number of data that I'm working with for that just keeps on going up. And I've got 3 different supervisors, and it's also in association with 2 different partnerships. So I've created a partnership with Melbourne Water, and I've also got a partnership with a SERA project, which is part of the Biomes of Australian Soil Environments. So firstly, I just wanted to talk about restoration in the soil environment. So basically what we know at the moment is that soil changes with disturbance, but we don't actually know what those changes are and how it relates with what we're seeing above ground. So we can see that based on WAG's literature, the soil community actually fluctuates in terms of diversity. And one of the first few things that actually change are the mycorrhizal communities, which is the yellow line indicated on the graph. But I suppose the main reason why we haven't actually looked at this previously is because soil biota was really hard to measure until technology got up to the level where we could do those genetic analyses that show what's happening. And now we've got other questions posed from that. Does soil actually determine restoration success? That's something which we still don't really know. There's a little bit of literature on it, but it's kind of connecting that with what's happening in our riparian ecosystem. That's a huge knowledge gap, and this goes to the overall aims and hypotheses for my project. So the role of soil microbial communities on the success of woody revegetation and riparian ecosystems is the overarching theme of my PhD. But within that theme, I found that there's heaps and heaps of different questions, and that's just because the riparian ecosystem within Australia in terms of mycorrhiza has been quite overlooked. And everyone's decided, well, this is an area with high nutrients, so why do we actually need mycorrhiza there? And maybe they have a point, but we don't know, so that's why I'm doing this research. But today I figured that because I don't have the mycorrhizal data, I'll tell you a little bit about the effect on restoration of microbial community profiles. So at the moment I think that microbial function will be lower at degraded sites. That's what the literature has shown. That's what we kind of think will be happening, but is it really what's happening? So the study outline is that I looked at 6 different waterways. So you can see all the waterways on the map there. So there were 3 on the east side of Melbourne and 3 on the west. But I'd also like to point out that we've got Main Creek at the bottom, which is kind of out on its own. Melbourne Water helped me choose the sites. So we chose them based on the fact that they're all really different. There's a lot going on there, and we wanted to know if the patterns that you see at one site are consistent across the 6 different waterways. And then within each of those 6 waterways, I've got 3 different treatments. So we've got our degraded treatment, which is basically a cattle farm, could potentially be restored, but we haven't restored it yet. We've got revegetated sites, which is 5 years or more in active revegetation, and our reference sites, which is remnant vegetation. And you can see that the different sites along the trajectory there. So in terms of my field sampling, I had a really intensive period in November, December last year, where I sampled the flora within 3 different levels. To sample the canopy, I used the point intercept method, and then subcanopy line intercept method, and then understory I used quadrats. This was within my 25 metre by 25 metre quadrat, so I sampled a lot of vegetation, huge amount of data for that. And then I'm adding that to the data which I collected for the CSIRO project, following their protocol with collecting soil at 2 different levels, 0 to 10 centimetres and 20 to 30 centimetres, just to see what's happening at the two different depths. And that involved collecting 27 cores. I think in total I collected about 2,000 or 3,000 cores worth of soil. And from that soil, I used it for physical and chemical analysis. The analyses which I was given were supposed to be really comprehensive, but when we looked back and started going through the data, we found that maybe they weren't as comprehensive as we thought, but that's something to look forward to in the future and figuring out where that fits in. And then finally, I've added to the base soil sampling protocol by including Ecoplates, which look at functional diversity. So I just wanted to explain what an ecoplate is. They were in the literature sort of 20 years ago, and they went away, and now they've come back because the genetic analysis is kind of up to date. We can say what we've got in function relates to what's happening and what species are in the community, or at least that's what I would like to do. So Ecoplates test for functional diversity changes over time. And so we've got 31 different carbon sources in a control within 3 replicates in each plate. And what happens is the tetrazolium dye has a redox reaction, and it changes colour if the soil microbes are using that particular carbon source. And in turn, that can be related to what's happening in terms of enzymatic processes within soil structure, the Krebs citric acid cycle, and nitrogen. And in terms of what I did to test this, you can use fresh or frozen soil. In my case, because the field season was so short and I wanted to make sure I collected everything in a very closed period of time, I used frozen soil, which I then dark incubated at 30 degrees for 120 hours in a low concentration of sodium chloride with some soil. And what I've found in terms of my physical and chemical analyses is that there's a difference in terms of the texture of the soil. So you can see here the degraded soils have lower amounts of sand, but then they've got higher silt and clay. And you can also see the chemistry of them so I'm only showing the data that has a significant result. The chemical analysis, the phosphorus was different, and I actually think this is based on the fact that some farmers may have put phosphorus amendments in their soil not long before I actually collected it. And I also found the main result and what all the other soil significant results fit with is that the pH is significantly lower in the reference treatment than what it is in the other two treatments. And then in terms of what happened with the function, I broke down the 31 carbon sources into 6 different carbon guilds. So the one which I haven't shown here is my control. There was no significant difference between my control, so phew, everything worked. And you can see here that function is always higher in the revegetation treatments, which is really interesting. And it goes against what the literature was telling me, so that's quite cool. And then when I put all this data into an NMDS, and it does its magic and creates an ordination in space of what's happening with your data. With the vegetation data, we can see that the physicochemical factors and vegetation data show quite a cool pattern where you can see the reference and revegetation sites are overlapping quite well, whereas the degraded site's off to the side, which is what you would expect, because degraded site, remember, it was just a patch of grass versus another patch which had trees, understory. There was a little bit more going on at the reference and revegetation sites. The other thing that's cool about this model is that it's actually a good fit, which was surprising. It's unusual to get a good fit first time you do something like this. And then that relates to what I found with the function. So when I do an ordination with my physical and chemical factors and my Ecoplate data, which is connected to the function, we find that the degraded sites are really constrained because they're less complex and non-specific. So what you're seeing visually is actually a great representation of what could possibly be happening in terms of our restoration objectives in the soil. So it looks like maybe we're moving towards something good, hopefully. And again, this model's a great fit. The stress factor fits really well, which I was quite surprised by. So some conclusions which we can get from this work so far. I decided to look at what's happening with the revegetated sites, is that they can be characterized by a lower proportion of exotic plant species. It makes sense. You plant native species, don't really want a lot of exotic species there. The proportion of clay is lower in revegetated sites than degraded. The pH is similar to what's happening in our reference sites, and phosphorus is similar to the reference sites as well. So those are all good signs. The other part, which is quite surprising and goes against what literature was saying, is that revegetation sites are more functionally active. We can speculate why, but at the moment we don't have enough data to say. I haven't shown you, but there are quite a few location differences. So as you can imagine, Main Creek is slightly different, but not always. I've found that some of the other sites are different in terms of vegetation. One of them had a major erosion event and everything just slipped away. And yeah, it just brings up the question of why do we have a different outcome from what happened in other studies? Which brings me to my future directions. So we're going to look at differences in composition, which I was hoping to show you, but I'm not quite there. And I've also got some data to look at a chronosequence across multiple ages of re-veg. And finally, I'd just like to go through, got a lot of acknowledgements and a lot of serious funding from many different bodies. So thank you for listening. Very broad. So if you have a look at the Bioplatform's website, you'll see that they've got specific markers which relate to, I think, so it's fungi, bacteria, archaea, and there's another group which I'm not so interested in, but I'll get all of that data and be able to analyze it.
So we move on to our last speaker today, which is Dr. Wendy Williams from this university, Queensland. So she'll be looking at microbial biobanking, so cyanobacteria-rich topsoil facilitates mine rehabilitation. Thank you.
Hello everyone, my name's Wendy Williams, and this afternoon I'm talking about cyanobacteria again, which is going back to Yolanda's first presentation, for those of you who are here for Yolanda's. And we in particular were a group of researchers who coordinated a project in South Australia at Jacinth Ambrosia mine, which was the Iluka Resources mine there. And so because cyanobacterial soil crusts and biological soil crusts were a major component of the landscape, they were needed to be taken into account in the rehabilitation project, which actually initiated our project. So with the post-mining restoration, we need to look at solutions to complex issues, so it's not just all about putting back soil and hoping that everything works, because there's quite a lot of disturbance, and our particular focus, as I said, was on the topsoil because of the fact that there is quite a suite of microbial communities there, and in this environment there was it was excavation, and so obviously there was massive disturbance. The Jacinth Ambrosia mine operated on low-profile topsoil stockpiles. So what they were interested in was the combination of what was there in the first place, what needed to be replaced, and what function it had. So we were doing a combination of studies to do with the biological soil crusts. So just to give you an idea of the location of the mine, it was in South Australia, right on sort of close to the Western Australian border, very low rainfall, very harsh environment, high evaporation, wind, erosion played a major role. So our aims were to actually look at the ecosystem function provided by biocrusts in the natural environment, but also to look at the effects of stockpiling on their survival, because we wanted to know whether or not they would survive the stockpiling process, because there's a lot of literature and a lot of work being done that shows the burial of biocrusts and burial of cyanobacteria results in their death or a longer term impact on them. And so that's what we were looking at as to what their survival rate was over time in the stockpiles. So the soil environment there consisted of three main soil management units, SMUs, where there was like a deep calcareous soil, a shallow one, and a sandy loam. So we focused our initial survey work on these three main SMUs just to see what was there, whether there were any differences, whether they needed to take that into account in their revegetation and so on. So that just gives you a bit of an idea of where our actual sample sites were in relation to the different SMUs that we went for and what they looked like physically in the environment. So what we found in this situation was that we found that there was different cyanobacteria across the different soils, because of course our study we focused on cyanobacteria, even though there were a lot of lichens and other components there because of the fact that they were early colonisers. And so although there was differences across the three SMUs, those differences didn't actually show up to be significantly different. There was different on a small scale. You could pick up a sample and look at it and find, oh yes, well this is dominated by a particular species, but on a broad scale there was actually no significant difference between them. So you could potentially find any one of those species anywhere across the mine site. On the similarity graph there, the TS2Y, I just explained, was actually our early study on a 2-year-old stockpile. So that one stood out a little bit from the rest in some of the survey work. So we just picked that up initially, where we just did some surface studies on that in order to just get a gauge of what was going on before we did the further studies on the stockpiles. So the cyanobacterial community structure was quite broad, the species richness, and interestingly among that there was a lot of nitrogen fixes, so obviously they played quite an important role in the restoration and the rehabilitation. We supported our physical microscopic examination and taxonomic evaluation of the cyanobacteria by sequencing and confirmed some of the different species that were there. So both studies sort of complemented each other, which is an unusual focus of the study. Normally you might go for one or the other, but not both. But in this case, because we really wanted to understand more about the community structure and the importance of it, and the importance in terms of rehabilitation, it was very important to actually get a good handle on what was there. So we found in the study, in the stockpile side of the study, that the cyanobacteria were moderately resilient to stockpile, but the species richness was greatest, and this picture at the background is the 2-year-old stockpile that we actually took some of the samples from the surface initially, but the species richness was greatest in the top 10 centimetres of the stockpile. But over time, which was really interesting of the experiment, because we actually did an experiment in the laboratory with the stockpile samples, that over time all of those species ended up coming back, but over a long period of time. So we concluded from that if you were in a natural environment trying to recover those species, then they would over time come back. But if you had long periods of dry weather and long periods of or drought, which often occurs there, then it could in fact make an impact. So the stockpiles results here show you where you've got a comparison between nearby the stockpiles, the natural environment at depth, and the stockpiles and the species, average species richness across the two biomes. But obviously in the undisturbed sites, naturally you're not going to have as many species at depth, because that's landscape that has not been disturbed for, like maybe thousands of years. When you're getting down to around sort of like 50 or 100 centimetres depth, it's quite deep as far as and in a totally undisturbed landscape, which this mine site was in the first place. So that was not unexpected in that sense. However, I think that it's important in this situation, the take-home message is the fact that the method that was used there, where they scraped shallow layers, so the initial scraping of the soils for the stockpiles was to a maximum of 10 centimetres deep, which is quite a shallow scrape for a mining process, and the stockpiles were, they tried to keep them no longer than 2 years. As you can see, there's one there, 29 months. However, as the process went on, they also did some direct return, and even though we don't have the actual data from that here, the direct return proved quite successful in terms of a fairly rapid colonisation on the soil crust. Now, what's really important about this, apart from the fact that the cyanobacteria are early colonisers, is that these areas, they're really prone to wind erosion, so even in stockpiling the soils, just having them, the cyanobacteria, actually recolonise the tops of the stockpiles themselves stops the wind from blowing it away. So what they actually did was that they watered them once, which created a chemical crust, an initial crust because of the salt content of the soil. But then the cyanobacteria grew really quickly that were in those surface soils, so it actually protected it. And then on top of that, they used the tree mounds and that to actually improve the cut-off of the wind as well. And then in their rehabilitation, it proved quite successful as far as the younger the stockpile, the greater that microbial community re-established. Unfortunately, I don't have time and don't have that data here to show you today, so I'll kept it short and sweet because I know at the end of the day we're all getting a bit tired. So I'd like to thank the team that we worked with from the University of New South Wales, the University of Queensland, Iluka and Jacinta Brosier rehabilitation staff. There was a lot of people who actually did this, and we are actually not far off publishing this work in biogeosciences. So it'll be good to actually understand a little bit more about the process of stockpiling and the importance of the microbial activity in it. Thank you. Thank you for. I shouldn't need reminding about this, but because I'm one of the coordinators, in 2019, in August, we are holding a biocrust conference on North Stradbroke Island, so nice and close here, at the Moreton Bay Research Station, a UQ facility, and that will cover a range of topics from just people who love biocrust to restoration. Yeah, there's a few people who'll be there that are in this room, but also to do with restoration, land management, soil microbial functioning, and a global perspective. The last biocrust conference that we had, there was people from over 26 countries represented there, from all walks of life, from mine restoration all the way through to land restoration and general, like the ecology of like ecosystem function, that type of thing. And so it's gradually grown from being a really small group of researchers to a really large group, and very interesting and well worth attending if you can. So if you can just jot down to email me at biocrust4@uq.edu.au, and I will add you to our mailing list if you're interested. And a website will be up soon, so shortly you'll be able to search it under the university masthead, and there'll be a website up as well. So please do come along if you're at least a little bit interested in those sorts of things, and we'll welcome you with open arms.
I just want to thank the presenters today, and thank you for keeping to time with questions. It made my job really easy, so thanks once again.